basic biosensor would be your typical thermometer that we used to have earlier, where you could place a thermometer in the mouth, read how much the mercury has risen in this calibrated scale, and as per that you would calculate the temperature of the patient or of yourself. And then of course we moved on to the digital era and everything became digitalized, so you have a digital output. So this is the display here, and you don't really have to see how much the mercury has risen. You get a basically there is a transducer here that gives you a quantitative value of that in terms of a display here. This is an LCD or liquid crystal display that you have. Then of course in the epidemic times you don't want to have touch related sensing, so therefore what you want to do is to basically check the temperature of individuals from a distance. So this is a non-contact infrared thermometer. So from a distance you give an infrared, and that basically the contact of the infrared with the body will allow it to detect what is the temperature there and give you a display here. This is an indigenous test. The test is called Feluda, and this is basically a CRISPR-Cas based strip based test developed by CSR. And the good thing is it could be a point of care test. Biotechnology, everything works at nano scale, so this is a nano biosensor again, right? And of course the first vaccine candidate launched into the clinical trials is an mRNA vaccine delivered via lipid nanoparticles. So again, nano biotechnology comes in everywhere, whether you talk of drug delivery, whether you talk of sensing, or anything that you can think of. Nano biotechnology is changing the face, and nanotechnology itself is changing the face of how things are understood, how things are done. For example, even your TV screen. So the plasma screens are now moving to nano based material. Likewise, if you want to talk of digital storage. So one of the major problems now is that we are generating so much of digital data. How do you store that data? And here again, DNA is one of the densest material for storage, and a whole lot of a classroom full of CDs can be stored in one molecule of DNA. So everything that you can think of now is coming to the nanoscale and as the biomolecules, because biomolecules have been doing this for millions of years, right? So this is now being realized that the best way of going forward in terms of sensing, in terms of storage of data, in terms of retrieval of data, is to go into the biomolecules, specifically DNA or proteins or RNA for that matter. So let's talk a bit of history. The father of nanosensors is Leland C. Clark Jr., 1980 to 2005, and he was the first to give the idea of biosensors and then also to use them and make some live displays. We'll talk more of this in later lectures. Then, of course, what is a biosensor? A biosensor is a device that detects, quantifies, transmits, and records the information on biological analyte or a biological process. So there is a biological analyte that you want to measure, which will basically give you an idea of a biological property. This biological analyte has to interact with a biological recognition element. This could be, so if this is a DNA, it could also be a complementary DNA strand. So the two hybridize, and the hybridization is the extent of hybridization is what you're going to measure, and that would indicate, whether these molecules are similar or not similar, or whether a certain type of gene is present or not present, and so on and so forth. It could be an antigen and an antibody. So you are suspecting that there is some sort of infection that is there. So you present an antibody on the recognition element, and then you give the patient's sample here. The patient sample, the patient blood cells, or let's say the plasma, will contain the antibodies. The antibodies bind to this, and the response that you will get here would also be in concentration. It will be concentration dependent. So based on the amount of interaction that happens, you'll have a signal that will be high or low, and that would allow you to basically quantitate, semi-quantitate, or quantitate, or qualitatively analyze what is happening. So that is important again. So you have you have an analyte. This analyte could be DNA, RNA, protein. It could be carbohydrate in case you're looking at the sugar levels. It could be a carbohydrate. It could be a lipid in case you're looking at the fat profile. So it could be anything, and then there has to be a biological recognition element that has a biological property of interacting with your analyte, right? And this interaction has to be concentration dependent. It must be able to report what is the extent of interaction that has happened, right? And that signal is basically the interaction that happens is basically deduced by a transducer. And this transducer functions then to convert it into an electrical signal. This electrical signal is amplified, and after amplification, you have a monitor that displays the result. And this again is basically, mostly it is quantitative in the sense it will give you a concentration or something per ml or value that is relative, and this value should be able to tell you whether your interaction is happening at a high level or a low level and what are its implications. That is also important. For example, you're measuring the glucose level, so you should be able to get what is the glucose level. That should be able to tell you also whether the person is diabetic or non-diabetic, hyperglycemic, hypoglycemic, and so on and so forth. So that is what is the basic idea of a biosensor. So our biosensor would consist of three major elements. If you see, one is the biological recognition element that is going to interact with the analyte. Second would be the transducer that will quantify this interaction and present it quantitatively in terms of how much of the interaction has happened. And then you have a processor. Processor itself can be of three parts. It could have basically a mechanism where the electrical signal is generated, then amplification is done, and then a display, which is important again. You need to be able to and quantify what is the extent of interaction that is happening. So that is typically a biosensor design. A biosensor is a device that detects, quantifies, transmits, and records the information on a biological analyte or a biological process. Example of analytes include nucleic acids, proteins such as enzymes, antibodies, or antigens, or other biological components such as glucose. A basic biosensor assembly includes a biological recognition element, transducer, and processor. So these are part of your biosensors: a biological recognition element, transducer, and a processor. And analyte is what you're going to give for it to sense. So this part is your biosensor, and here is your analyte that you would give so as to identify what is the interaction that is happening. The transducer converts a molecular biological signal into a digital or electrical signal proportional to the concentration or physicochemical spatial properties of the analyte. So this would be in terms of concentration, in terms of the amount of interaction that has happened, that it is going to convert and can be amplified, qualified, displayed, and analyzed through a processor. Right? So this is the whole basic design of a biosensor. Any type of biosensor you will have, it will have these elements. So it will have a biological recognition element, it will have a transducer, and then of course it will have a processor. Right? And then today we'll talk of nanopore, which we have done already in the initial classes again. And there, of course, you do not have basically you have a recognition element, but it is not in terms of actually interactions. We'll talk of that. So this is another alternate way of representing the biosensor design. So you have biological material, you have biologically sensitive element or biological recognition element. Basically these two will interact, and then this interaction is quantified by the transducer, converted into an electrical signal, and then you have a display, and you could have a reference material so as to check whether the reading that you get is correct or not. So the reference material, you will have a known reading, and therefore you can basically, if the known reading is found to be correct, then you know that the sensor is working fine. Now an untested material can be tested. Also sometimes reference could be what is the base reading that you get with respect to the reference sample. And this is important again, signal transduction. You have the biological element that will interact with your analyte. Then you have a biological response. This will be quantified by the transducer, converted into an electrical response, and then that measurement will be displayed to you. And here is again the basic design of a biosensor. So you could have an analyte. Analyte could be anything. You have biological recognition element. Biological recognition element could be basically a part of the biosensor that is either aligned by an antibody. So antibody would be specifically bind to an antigen, and this interaction can be quantified. It could be an enzyme. So you give a substrate, the enzyme would convert the substrate into a product, and basically you could actually be assaying what is the product that is formed. So the property of the product is what you're trying to assay. It could be a microorganism, and you could give a specific receptor that is specific to this, and that could give you again a signal. Then of course it could be a DNA, and this could be what is shown here is a double-stranded DNA, but actually it will be single-stranded, and you'll basically will give the other DNA as your analyte, and if the two interact, basically if the two are complementary, they would interact with each other, and that can again be given as a signal. You could be labeling one of these strands with a fluorescent probe, and we'll talk of more details of how labeling and everything is done in the coming lectures. And I'll specifically give you an example of microarray in some detail, so that you know how this is done and how microarray can be used for understanding gene level expression, and it is done at a high throughput level. So what is also important by technology now is that we're moving from one gene, one protein, etc., to high throughput and understanding of things. High throughput means if there are 11,000 genes that are getting expressed at one cell in a specific time, all 11,000 have to be, you know, their expression levels have to be determined in a single experiment, because genes are not expressing in isolation. They are impacting each other in several ways. So therefore, what you need to understand is how many of these genes are active and at what levels, and then you can possibly understand that, okay, if gene A is active, therefore it also you know, activates gene B, which becomes active again, and so on and so forth. So you're trying to deduce relationships, or you're trying to do what is known as systems biology. You're trying to understand as a whole how the cell is interacting, and therefore it is important that you basically try and understand the entire components of things. And therefore you have genomics, the study of entire DNA, not just the genic part. Genic part is only 1.5% of the genome. What you say is what is also the remaining 98.5% doing, which will have regulatory regions, which will have control elements, which will have repeats, and so on and so forth. So you have genomics, then you have proteomics, where you want to understand the entire protein expression profile of a given cell at a given stage. So this is basically, again, space and time specific in a specific organ in a specific time. What is the extent of, what are the components of proteins, entire number of proteins that are getting expressed. So, and what you basically want to do then is to compare a normal cell versus a disease cell, and then find out which of these proteins are differentially expressed. There will be some proteins that will be expressed at a lower level in normal cell, and then there will be some proteins that will be expressed at a higher level in normal cell. So you want to basically try to find out, you're trying to find out what are the protein components, or what are the proteins, or what are the genes that are differentially expressed between normal and diseased condition. That would then give you an idea of how many genes are involved in the phenotype that you're looking at. So here you are. If you see, this is a biological recognition element. You have an antibody, you could have an enzyme, you could have a microorganism, you could have a DNA, you could have other cells. These would interact with an analyte, and then these would generate a response that could be electrochemical, that could be optical, that could be mechanical. Electrochemical itself could be several types. It could be potentiometric, so the potential changes. It could be ampemetric, where the electric current changes. It could also have a piezoelectric effect. We'll discuss that in more details. Likewise, it could generate an optical signal. Optical signal obviously would be in terms of release of a certain, you know, colored wavelength, and that can be recorded again. We'll talk of this in more details today itself. And then, of course, there could be mechanical signal in terms of increase in pH, increase in pressure, increase in temperature, whatever. All of these signals are converted into an electrical signal, and then they are amplified, and then they are displayed here. So this is basically a more comprehensive diagram of a biosensor. If you see, we are talking of what type of analytes you can have. You're talking of what type of elements could line up at the biological recognition element. You're talking of what kind of signals you can get here. It could be electrochemical, optical, or mechanical. Then you're talking of how the signal is transduced and quantitated, amplified, and then, of course, how the data is acquired, processed, and displayed finally, right? So a biosensor is a device that detects, quantifies, transmits, and records the information on a biological analyte or a biological process. Examples of analytes include nucleic acid, DNA, RNA, proteins such as enzymes, antibodies, and antigens, or other biological substances such as glucose. And then, of course, a basic biosensor assembly includes biological recognition element, transducer, and processor, the output system that is there. And then, of course, a transducer converts a molecular biological signal into a digital or electrical signal proportional to the concentration or physicochemical spatial property of the analyte, and can be amplified, quantified, displayed, and analyzed through a processor. So this is basically a complete definition of bioprocessor. And here I show you one biosensor here, which is basically antigen-antibody interaction. And this antigen-antibody interaction is then converted into a signal via the transducer, and then you get your data here. The other second type, which I show you at the bottom, is basically based on hybridization and also based on fluorescence. So if you see, you have this fluorescence mark here, and fluorescence basically is what you're going to record here. So this is an optical transducer, and in this case here, this antigen antibody interaction, it could also be an optical transducer if your antibody is having a fluorescent tag, or an analyte is having a fluorescent tag. It could also be based on what is known as surface plasmon resonance. So surface plasmon resonance is a phenomenon where there is no label. So you will talk of more details of that again in the next lecture probably. So we'll keep it for that. But for now, you understand that this could be optical, this could be again optical again based on what is fluorescently tagged. And then of course, here you're looking at hybridization. So if you give a complementary DNA strand, it will go and bind, and therefore you will have a bit of fluorescence. And we'll talk of each of these in more details in coming lectures. All right, class. So here you are, nano biosensors. What is the advantage? Why are we moving towards nano biosensors? So nanostructured materials-based transducers enhance the sensitivity by more than one order of magnitude. So the important thing is that the sensitivity of the machine or the sensor as compared to a conventional sensor is much more when you use nano biosensors, up to 3 orders of magnitude highly more sensitive, which means even with the lesser amount of analyte, or even when the cell is expressing very small amounts of your marker, you will be able to detect a particular physiological or pathological condition, which is again important when you're talking in terms of fighting the disease. And if you remember, when we're talking of cancer diagnostics, I had shown you how quantum dots are almost 50 times more brighter than as compared to conventional dyes. And even at the lesser expression of the biomarker on the cancer cell, they are able to detect and give you a signal. So which allows you to detect the pathological state early in the earlier stages. And that is very important in terms of diagnostics, and also in terms of prophylactics, and also in terms of the remedy that you are going to suggest. In cancer, one of the major problems has been that it is identified, a person appears absolutely normal from the phenotype, and only at a later stage you get to identify that the cancer has progressed, and that is a major problem. So in that sense, nanobiosensors, because of their enhanced sensitivity, can come in very handy in identifying the disease much earlier, and even and at a point where it can be controlled, right? And this improved analytical performance is attributed to factors such as better electrical communication ability of nanomaterials. So we know that at nano scale, the properties of materials are very different from the bulk scale, and at nano scale they are basically highly conductive. So therefore, the electrical signal can be much stronger. They are also highly the magnetic properties, the electrochemical properties, their binding capacity, their catalytic properties, all are enhanced, and therefore at nanoscale you have higher sensitivity than the conventional material that you have. So high loading of recognition elements, so they can bind to the recognition element at much higher efficiency as compared to what it was with the conventional non-nano sensor, and therefore what you get is higher specificity, higher sensitivity, and also what is important is that you get to miniaturize the machine. So now your machine is so small and so handy that it can be taken to the patient instead of patient coming to the machine. So that gives you greater portability, and therefore this also gives you an option of being a point of care technology. What is also important is that a diagnostics goes to the patient instead of patient coming to the diagnostic center. The diagnostic machine should go to the patient, and that will allow you higher coverage and allow you a better response in conditions such as epidemics and pandemics. And therefore, again, nano-based biosensors are very important. And we'll take one example today, which we've done earlier also, and then we move on to other things. Right? So here you are. We have done this earlier also. If you remember, this is MinION or nanopore. This is a sequencer of the size of a pen drive, and that can allow you to sequence the DNA. And now also you must remember that sequencing is now the new microscope. There were times when you used to do microscopy to see what is the infection and everything. Now for everything, sequencing is becoming the first level diagnostics and the final level diagnostics. And therefore, this is very important. that you have a sequencer that is so small that can be held in hand, that is so easy to operate, that with very basic minimum apparatus you are able to operate it. So this is a comparison of how nano sensing can miniaturize the machine. These are your normal sequencers, Sanger sequencers, some of you may have seen. Now of course they are obsolete and moving out, because now we have moved to next generation sequencers. Pyro sequencer was also obsolete, so it came in 2005 and now it is obsolete because the technology has changed. These are the current sequencers. Illumina sequencer, which is basically your virtual terminator sequencer, and it is the most successful sequencer that we have in the market today. And then you have Ion Torrent, which is a successor of Pyro sequencer, and this is also being used in many labs. And of course, the best option that you have is the nanopore, which is miniaturized, which can be used easily. And the advantage as compared to other sequencers is that this is a long range sequencer. So it gives you long read lengths. So starting from one end, you can read up to 20 kb of sequence, 30 kb of sequence, 60 kb of sequence in some cases, which is important. And we'll talk of that, why that is important in a moment. So this is your biological nanopore. If you look at if you look for inspiration into biology, whether you can have a nanopore in biology. So there are known proteins that can serve as nanopores. If you see here, this is alpha hemolysin. This is a heptameric protein. 7 polypeptides come together to make this protein. And it has an inner diameter of about 1 nanometer. So this is within your nanoscale. And this can be used as one of the biosensors, and it has been used as a biosensor. And now, of course, we have synthetic biosensors as well. This diameter is the same scale as many single molecules, including DNA, right? And what people have done is, how this technology is being used for sequencing is basically, if you place a nanopore in an electric field, there is a basal level of current that flows through the nanopore, and you will have a stable current or at certain amperes of range, and you can record that ampere, and you know that the basal conductance is this. In terms of current, it is these many amperes of current that flows. Now, when you pass a particle through the nanopore, because of the stereochemistry and the shape of the molecule that is passing through, there are characteristic fluctuations in the current that happen, and those fluctuations basically allow you to detect what molecule is passing through. If you have a reference, you have already checked that when you place this blue molecule, this is the amount of fluctuation that happens. So next time you take an unknown molecule and you see the fluctuation is the same amount, you know this is the blue molecule. So here I show you 3 molecules here: red, blue, and green. Each one is producing its own characteristic current. So now if I have a untested molecule, which is one of these 3, I can allow it to pass through, and I can know that, okay, if this is the fluctuation that happens, then it is the red molecule. If the fluctuation is slightly more, it is probably the blue molecule. If the fluctuation is intermediate, it is probably the green molecule, and the green molecule is big enough not to pass through the nanopore, if you see here, right? So this is the basic idea. It is very simple, very innovative. The idea is simply to allow a molecule to pass through a nanopore and check for changes in current. Now, with respect to sequencing, why this is important is because adenine, thymine, cytosine, and guanine will give you different fluctuations as they pass through the nanopore. So that is the whole idea. And based on this fluctuation in the current, you are able to deduce what is the nucleotide that is passing through. Now, this is important because in 2 ways. One, until now, what we had been doing for sequencing was to make a complementary strand with DNA polymerase and check for what is the nucleotide that is getting added, right? And based on that, you are deducing the sequence. Here, in this case, you're not synthesizing a complementary strand. You're simply forcing a strand of DNA through the nanopore, and as it is moving, you are simply checking for changes in fluctuations of current. Because A, T, G, and C give you different fluctuations, you are able to record and identify what is the nucleotide that is passing through, and that gives you the sequence. And because you're not using polymerase, you can allow a very long strand to pass through the nanopore and record what is the current fluctuation. That is why here you have very long read lengths, right? Which is important, because polymerase normally, it cannot, if you use a normal Taq polymerase in lab, you cannot amplify more than 1.5 kb of sequence. So wherever your primer is, from that start point until the next 1.5 kb, at max 2 kb is what it will give you, and then beyond that, the processivity is not there. It will fall off, and therefore there is a limitation to what length you can read when you use a DNA polymerase. But in this case, because DNA polymerase is not being used, you have a very long read length that you can get, right? All right, so we move on, and we had done this earlier also, so I think this is just a revision. We get into fluorescence in a moment now. So what is sequencing? Sequencing basically is to determine the order of nucleotides. And why do you want to determine the order of nucleotides? You want to determine the order of nucleotides because you want to know what constitutes a human genome. Second, more importantly, what you want to identify is the variation. So how much is the extent of variation that will allow you to remain human? And more importantly, we are looking at what are the variations that cause disease. So we are looking at inborn errors and inborn genetic defects or genetic diseases. And we're looking at what is the difference between a normal individual and a person who suffers from a genetic disease by comparing the sequence. So we're looking at variations which are specifically associated with disease. And if you want to do a genetic engineering, or you want to do a basically genetic engineering and correct the error, then it has to be, you have to know where the error is in the first place. So that is what sequencing allows you to do. Sequencing will tell you what is the basic composition of ATGC starting from chromosome 1 until chromosome Y, what is the nucleotide sequence that is there. When you compare these nucleotide sequences with known mutations, you would be able to identify whether the individual is predisposed to a certain diseases or not, and what precautions he can take to basically, try and avoid getting into these diseases. So some metabolic disorders, you can take precautions and not take a particular type of food, and that will allow you to, be safe from a particular type of allergy or a food intolerance or something like that. So therefore, in that sense, it is very important. Also, it tells you, what is the potential of you developing a certain disease based on your family history, based on the mutations you carry. So that is important. Here are some of the sequencing milestones. This slide I show you new. This was not there earlier. So if you look at the sequencing milestones, the first sequence was the first bacteriophage sequence way back in 1977. This is 5X174. Then, of course, Haemophilus influenzae was the first free living organism to be sequenced. And then, of course, C. elegans was the first multicellular organism to be sequenced. And then the crowning glory of Sanger sequencing was 2001 when the human genome was sequenced. And this took around 10 years and, to $3.2 billion. So almost $1 a nucleotide to sequence. And that is when you started with the second generation sequencing technologies, because you knew that this is not the technology that is going to take you forward. And what is our objective in sequencing? We want to move to what is known as personalized genomics or personal genomics, where we want to give the individual a tailored medicine which is customized to his requirements, right? And therefore, for that to happen, each individual has to be sequenced first. And if each individual to get sequenced will take 10 years and $3.2 billion, it is not happening at any point of time. And therefore, you started with your quest for next generation sequencing technologies that could give you a sequence, a complete human genome sequence in around $1,000. And luckily now we are in that range, and you can sequence the human genome at less than $1,000 now, courtesy nanopore sequencing and other sequencing techniques. Then, of course, Neanderthal you also sequenced, and these are some of the reviews that you can go and refer back. These are very nice reviews, and that will give you an idea of how sequencing is today. All right. So as I was talking of read length here, this slide also you have seen earlier. So if this is one photo that I took of Eiffel Tower from one of the important points in Paris. So this is basically the Eiffel Tower, and if I were to tell you a challenge to break this into multiple pieces and then rearrange it together, and this is into 4 pieces, 9 pieces, and this is basically 56 pieces if you see. So you can see very clearly that if you were to shred this into these many pieces and rearrange, it will be easiest to rearrange here where there are long fractions, right? When you're making it into 4 fractions, so you have very large pieces. To rearrange them together is much easier as compared to rearranging 9 pieces. And this is, of course, much easier as compared to rearranging these 56 parts, which will be really, really difficult, right? Likewise, for genome sequencing, as I told you, because you cannot start amplifying from one end to the end of the full end of the chromosome, you basically break the chromosome into parts and then sequence and then put them together. So read length basically is the maximum sequence length that can be obtained in one sequencing reaction. So depending on what technology you use, you have to break your genome into as many smaller pieces. So basically, if this is your genome here, you will basically be, you should have, because you cannot start reading from one end until the other end, you have to break it into smaller fragments. These fragments are then sequenced. So now you have the sequence of A, you have the sequence of B, you have the sequence of D, you have the sequence of C, and you have the sequence of B. Now you have to rearrange them in the order they are present in the genome. And therefore, read length becomes important. So it is like, again, breaking this photograph into 56 fragments or 3 fragments or 4 fragments or 9 fragments. So the longer the read length, the easier it is to assemble back into the original order. So that is the whole analogy that I'm giving you here. So basically, the steps involved include creating a genomic library. You split the genomic DNA into smaller pieces. Then you sequence these smaller pieces because these are of the length that you can sequence given the technology that you're using. So you get, if you're using Illumina, you'll get around 100 base pair or 150 base pair of sequence. If you're using Nanopore, you'd get 20 kb, 30 kb sequence, and so on and so forth. So basically, that is important again. And then once you have these fragments, you have to reassemble it in the order they appear in the genome. So genome sequences, so while you have the complete genome sequence here, when you sequence the fragments, you do not have the order. So that part is known as assembly. You have to arrange the reads in the order they appear in the genome. And that is again important. And therefore, long read sequencers are very important. So if you see here, read length is the maximum length sequence that can be obtained in one sequencing reaction. This is specific to each sequencing technique. Read length is a critical parameter for completeness and accuracy of the genome assembly. What you're using here is basically a nano biosensor, which is basically your nanopore, which is put to an electric field. And that is basically when you're putting your DNA through it, you are identifying what is the change in current. So your transduction mechanism basically identifying the change in current, and that is then digitally converted into your sequence. And here are some more reviews that you should be reading so as to be updated with what is happening at the sequencing front. You can go back. These reviews should be available. If they're not available to you for free, you can write to me. I have these, and then I can send it to you for